Okay. Hi, everyone, and welcome to this Emergent Session of the Open Science Room. My name is Stefan, and I will just be in the background. And it's a pleasure for me to introduce Andre Marquand, and he will be doing a lot of talking from here on. So if anyone has problems, just tag me. I'm the OHBM Open Science SIG person. But otherwise, I'll just be in the background. And with you, Andre.
Okay, thank you very much. Welcome to this Open Science Room session. I'm very excited to present with you today. I have some people helping, some people participating in the session today. We have Sage Rutherford, hi. Mariam Zabihi, and we have some participants online. So Johanna Bayer and Charlotte Fafaza. And together we're going to talk to you a little bit about some of the concepts behind normative modeling, and we're going to walk you through an analysis or show you some resources that will help you get underway with your own analysis using normative modeling, the toolkit that we've developed, which is all available on GitHub. So I'm just going to start by sharing my screen. And so I'll just give a little bit of a conceptual overview to start with. And if there are questions, we hope to have enough time for questions at the end. So you can either put them in the chat or send them to us via Twitter. Any of the participants would be happy to answer your questions. So I'm going to start with a little bit of a mission statement, if you like. So this is really what we are about, developing innovative machine learning methods, applying them to big data, aiming to have impact on problems that really matter. I'll put some acknowledgements up right at the beginning, just to really emphasize that this is really a team effort, the development of these tools, both from the conceptual level, the implementation in terms of the underlying statistics, and also the deployment to software tools that we hope will be useful for the community. So this is really a team effort. The starting point for normative modeling really is thinking about how analysis is typically done in practice. In many, and I usually start with a case control paradigm to illustrate the difference between normative modeling and classical approaches. And I'd generalize that slightly to mean just both looking for group level differences in a classical sort of t-testing scenario, also for looking at continuous differences in terms of things like GWAS or classifiers. Really, essentially, there's some background noise. I think maybe someone can mute the microphone. Thank you. All of these things, the essence really is that they aim to detect group differences, which is part of the story, but I'd like to show you some results that indicate that it's not the whole story. So the first sort of classical departure from that framework might be something like a clustering approach, which aims to partition this moves away from this sort of arbitrarily simple scenario where there's clearly defined cases and control groups, to saying, well, actually there might be something akin to subtypes within the population. Now that idea has a very long history in psychiatry, and this is a slide from one of Tom Insel's papers to show that this idea of taking these heterogeneous diagnostic entities, lumping them together, acquiring data, and then trying to find strata that more faithfully represent the underlying biology has been done, in fact, for decades, and it hasn't really converged on anything that challenges the current nosology. And I think one of the reasons for that is because that this kind of movement between biological measures and reclassifications is not really well defined. And there's many other, one of the things that I like to suggest as a reason is that there's many things that could be going on within these clinical cohorts beyond the simple scenarios that I've outlined at the beginning. So normative modeling is one approach. I'm not saying it's the only approach, but it's one that we think is quite promising. From a conceptual perspective, it's very simple. It's akin to the use of pediatric growth charts, which you're probably familiar with. And the idea is that we map centiles of variation across the population in terms of some biological measure, for example, weight or height as a function of age. And so what we do in normative modeling is we just substitute these variables for things that are relevant to psychiatry. We can do this in a regional fashion to say, well, generate a growth chart for the brain, if you like. Also using other measures besides just age. So we might estimate brain behavior mappings, for example. By doing this across a large cohort, we can make predictions at the level of the individual, so statistical predictions about where each individual lies with respect to the population reference. And by doing this across a large clinical cohort, we can then use this as a basis for a subsequent stratification, for example. I'll just show you very briefly some work that was done by Thomas Wolfers and Mariam Zabihi principally, and I'd just like to illustrate that really this notion of detecting group level differences is really distinct from what we can observe at the individual subject level. So it doesn't matter what disorder, which is psychiatric disorder that you consider, we see whether there are robust group level differences. These don't necessarily recapitulate the individual deviation. So this is showing a map which has thresholded at 2%, meaning that any regions that you see have deviations in more than 2% of subjects. So you can see that there's, in schizophrenia, for example, there are robust group differences, but that doesn't necessarily reflect what's happening at an individual level. The same is true in bipolar disorder and ADHD, and even in autism, for example, where there are in fact very few case control differences. We see very widespread individual differences that are very idiosyncratic in the sense that they only happen in a subset of individuals. You might think that this always happens, and in fact that's not the case. So in dementia, for example, we see a much higher overlap in terms indicating a higher consistency of individual differences that has a closer correspondence to what you might expect in a case control scenario. So then we can use something else that that is a very important route forward in this kind of line of thinking is then, what do we do with those individual deviations? So we might take them forward into developing individualized risk scores, for example, something akin to brain fingerprints. We could develop by factorizing these matrices. So if we represent these individual deviations in a matrix, we can get some kind of factorization of these deviations, which give us sort of overlapping profiles. We could use them as a basis for clustering, and then this is a paper that Mariam produced and showed very good. Well, I mean, I won't go into these in detail. You can read the paper if you're interested, but it shows that why you don't see differences in autism, because you have both increases and decreases in different sets of individuals, which map onto symptoms and genetics. So how do we do it in practice? So that's a very high-level overview of some of the reasons why you might want to consider using normative modeling. There's a whole bunch of different algorithms that you could use. A lot of the initial work was based on Gaussian process regression, which is quite flexible, but it has some limitations, which we'll talk about in a moment. But there's other approaches, and many of these implemented in the toolbox at Sage. We'll demonstrate a little bit later on. At a conceptual level, there's a number of desiderata, if you like, things that these algorithms ought to do. First of all, we need them to scale to very large data sets, because we want to model variation across large population level cohorts. We need to be able to, they need to be quite flexible in the sense that we need to be able to model both nonlinearity and non-Gaussianity in terms of the stratification, in terms of the centiles of variation across the population. And we have to accommodate side effects, and that's something that Johanna will discuss in some detail a little bit later on. I'll just pause for a second there, because the original plan was that Charlotte would take over from this point. Are you able to do that, Charlotte? I don't see you in the presenting room, which was a bit of an oversight, perhaps. No, just continue. All right, okay, so I shall do that. Can you still see my screen? Please, I can't see both at the same time, so I'm assuming that that's okay. If not, please come in. Right. So the idea, the classical approach would be to use a probabilistic regression model, and this is many of the applications that I've shown you. I'm not going to go into the details, but basically the point is there is a severe computational bottleneck in using this approach, which relates to the inversion of this covariance matrix, which will scale very poorly in increasing numbers of subjects. So one way forward is to we still need to be able to model nonlinearities, but we can do that quite efficiently using a linear model. So using a set of linear basis function, nonlinear basis functions, we can use a linear model to model these nonlinearities in the curve, in the centile curves. And we can use a technique known as likelihood warping, which effectively involves applying a monotonic transformation that maps non-Gaussian signals to a latent Gaussian space, and that is by construction invertible. So we can string these things together in a very efficient manner, and it provides us a flexible way to model non-Gaussianity in the data. So this was Charlotte evaluated this in UK Biobank. We have excellent computational scaling relative to the Gaussian process model, which is really good. This approach really scales to population level data. And you can see this is an example using a particular phenotype from UK Biobank. These are white matter hyperintensities. You can see using a Gaussian model, you arrive at centiles which are inappropriate, whereas using this likelihood warping technique, it matches the underlying distribution much more closely. And that can also be used in a longitudinal fashion so that we can detect subjects that have a faster than normal aging trajectory, for example. Another big challenge is dealing with side effects, and as you can see, this is a large data set that was assembled by Sage. Just fitting some kind of centile curves to this distribution in a naive fashion clearly arrives at centile curves that are nonsensical because we don't account for side effects properly. So there was some work that you might just think, well, let's apply some kind of data harmonization technique to these data before doing that. We don't think that's really the optimal approach, and I'll illustrate why. This is work from Mosi Kia, and then I'll hand it over to Johanna, who'll provide an overview of her work addressing this problem. So supposing we have a number of sources of variance in the data. So these are illustrated here by these circles. So we have, for example, known variance that we measure. That might be something like age. We have interesting clinical effects that we don't happen to measure, for example, subtypes. And there's batch effects or site effects in the data, and these things can be correlated, as you can see by overlaps in these ellipses. So what most harmonization techniques do is something akin to the following. I've written combat here, but this is a problem in general. Is first we would remove the known variance from the data. That's the age-related variance. Then we would remove the site effects, and then we put back the age effects back into the data. And there's two problems with that. So first of all, we remove this part of the variance when we shouldn't, and it's very difficult for us to deal with this part of the variance that's correlated with the data, with the effects that we do measure. And so I will stop there. So if Johanna, if you're in a position to be able to take over and give us an overview of your approach that to solve this problem.
Yes. Just find my slides. Can you see my screen?
Yes.
Yeah.
Okay, cool. Let me just. Yeah. Okay. Yeah. So I'm going to talk about kind of the side effect problem, and this is part of my PhD, and the topic is accommodating site variation in neuroimaging data using normative and hierarchical Bayesian models. And the idea behind this was really like, as Andre said, that we discovered that these multi-site effects in neuroimaging data are confounded with variation of interest, for example, age and sex variation, and that it's not really easy to get this information, like to remove this side effect, the side effects from the data, because they're kind of confounded and you might take out a variation of interest. And I've plotted here the side effects in the Abide dataset. So this is the Abide dataset is kind of data derived from free surfer population. So this is just each box plot here is the cortical thickness of 34 regions on the cortex for each individual. And what I did is I kind of colored those individuals, like I colored them by site, and like within site, I ordered them by age. And you see like that cortical thickness decreases with age, but you also see like these shifts between sites. And you don't really know whether this is a side effect or whether this is whether different sites have just different age ranges. So that is basically what we mean with that there's a confound between side effects and biological variation or variation of interest. So now the idea that we had is that we could include site as a predictor in a normative model, because everything that you include as a predictor in a normative model, basically you get set scores that are then normalized by this predictor. So if you include site as a predictor in a normative model, the Z-scores that you get are not only normalized by age and sex, like if you include those two, but also by site. And my approach was to use hierarchical Bayesian regression in STAN. So STAN is a probabilistic programming language using Markov chain Monte Carlo to map the normative variation of cortical thickness with age, sex, and site. And like I said, I used the Abide dataset, which is actually an autism dataset, but it comes with 20 sites and contains information about 539 individuals with autism spectrum disorder and 573 matched healthy controls. And the information that I used from the Abide datasets is kind of what we get out of brief surfer population, so just cortical thickness measures of 34 bilateral regions, and based on the Desikan-Kiliani atlas, and then an average over all these 35 regions. So in total, I had like 35 measurements per individual. And I trained and created this normative model, including site, age, and sex to model cortical thickness for each region. I trained that on a training set of healthy controls, and then tested it on a test set of healthy controls and on the individuals with autism in the Abide dataset. And now a bit more detail to the model. I used basically hierarchical Bayesian regression, and I tried to kind of model my true value y using the predictions from the model F, and then add random noise on the diagonal. And this kind of the predictions F that I get from the model. So the model is basically a linear mixture model. So I have a matrix where I have like biological variation, so X, age, and sex, and a vector beta that kind of collects the estimates of those for this matrix. Then a matrix Z where I have like kind of a site index, and then a vector U where I collect like the estimates for the side effects. And so what we did in our approach is that we modeled random intercept offsets for sites, meaning that all these site estimates had a shared prior. And then you see like this parameter gamma, and like I created two different versions of the model. So in the first version, which is just a linear model, I set this parameter gamma to 0. And in the second version, this parameter gamma is basically a Gaussian process model. And like I have this Gaussian process kernel between all individuals. And then this is to model nonlinear effects between age and cortical thickness. So basically, I have a linear version of this model and a nonlinear version of this model. And then I estimate the three parameters of the model, which is the vector beta, the vector u for side effects, the length scale of the Gaussian process, and then aleatoric and epistemic noise. And I can then calculate the z-scores, like this is the kind of the equation at the bottom, and by kind of setting the difference between the prediction from the model and from the true value, and then divided by the noise and the predictive variance. And so now I wanted to see how these z-scores that I now get from this model, where I have side as a predictor, are free from side effects. But now I want to compare them to other ways of clearing the data from side effects. So what I did is I created a second normative model without side as predictors, so only age and sex as predictors. And then I took the data and cleared the data with combat, with age and sex preserved, and regressed outside. And I took like this kind of data that I harmonized and fed it through this normative model without side as predictor. So I also got z-scores, and I was now able to kind of compare the z-scores from a model with side as a predictor to the z-scores of a model where I had cleared the data in other ways. And I evaluated the performance of the model using different performance measures, evaluating means and variance. And the idea here is that these measures kind of show how compare the match between the true and predicted values. And the idea is that if they match, then the z-scores can be trusted and are clear of side effects. So what I use is the correlation between the true and predicted value, the standardized root mean squared error, the mean standardized log loss, explained variance, and predictive variance. And what I found is that for my model, so the ones where I modeled side, I had higher correlation between the true and predicted value, lower standardized root mean squared error, better MSLL, and higher explained variance. And I think the most important finding was that what you can see in this kind of graph on the right-hand side is that we had a significant shrinkage of variance for all but my models. So what I plotted here is like on the x-axis predictive variance, and on the y-axis the true predictive values, and on the y-axis true values. And you see that for my models, for the hierarchical Bayesian Gaussian process model and for the hierarchical Bayesian linear model, the dots kind of are on one line. But for all the other models, you see that the predictive variance is restricted. Which kind of tells us that these models basically do exactly what we do not want them to do, which is that with the side effects, they take out variation of interest. And I also found that there was no significant difference between the linear and the nonlinear version of the hierarchical Bayesian model, which is probably due to the age range that we had in the nearby dataset. And then I did another sanity test. So the idea behind a normative model is that if you train it on healthy controls and test it on healthy controls, then exactly 5% of individuals should have z-scores larger than 1.96 or smaller than minus 1.96, because that's the kind of that means like the 97.5 kind of percentile, and that means that 2.5% of individuals should have, like, on each side should have extreme values. So you see in the upper panel, kind of the testing the model on healthy controls, and I thresholded it at 5%. So you see, like, and kind of plotted it on the brain regions, like across the brain, and you see that the kind of number of individuals, or the percentage of individuals which have like z-scores of like plus minus 1.96 is roughly around 5%. And then in the bottom kind of panel, I did the same for autism, and you see that there is significantly more individuals who have atypical z-scores, or like z-scores of plus minus 1.96, larger or smaller than plus minus 1.96 as a z-score. And like the idea here was not to kind of look into autism or investigate autism, but this is more like a sanity check. We wanted to see whether the model can still retain clinically relevant information, and kind of it seems that it can. Then currently I'm applying the model to the Enigma MDD dataset, which is a very large dataset, because the idea is, like Andrea has already indicated, that if you train the model on healthy controls and then test them on like individuals with any kind of interesting feature, then this is kind of, you get individual deviation scores that kind of tell you about this individual feature, for example, about depression. And for this data set, I had to recreate or like rewrite my model, because I used this Gaussian process, kind of as nonlinear version of the model. And as Andre said, it's very computationally costly. So I had to create another model, which is a spline model. And now we're running that on the Enigma MDT dataset. And I'm also working on prediction of unseen sites and an application to voxel-based morphometry with Tim Han, who was really interested in that. And if you want to read this up, like I've written it up in a preprint, and also I have a poster with the number 1569 at OHBM. Yay! That's it.
Great, thank you very much, Johanna. That's really nice work. Would you like to respond? There's a question in the chat about whether this will be available. Will the scripts be available?
Yes, they are. They are available with the preprint already now. So if you look at the preprint, there's a link to my GitHub repository where you can find all the scripts. And shortly, there will also be a version that you can run in Singularity, because Stan can sometimes be a bit tricky. But yeah, there would be a containerized version.
Great, thank you. And then there's a question from Fidel about this preprint does not include the Enigma tests, right?
No, the preprint, no. But that will be, I'm working on that. Like I'm working on writing that up at the moment. Okay. That's my second paper, yeah.
Great, thank you. And I'll just also add to, I think it's really, particularly the scatter plots for me are very convincing. And I'll just also add that there's a sort of a slightly different but parallel implementation available in the toolbox that we're going to demonstrate in a minute as well. So thank you very much, Johanna. And I will just share my screen here. So I have a couple of points that I would like to just add to that. So indeed, and this was something that you touched on in the last slide. So indeed, how to apply this to new sites is a very important problem, and we've approached this in a federated learning manner. This is not really the place to go into a detailed discussion. I'd encourage you to read these two papers if you're interested in how this works. But the idea of that we have these very large samples that are perhaps difficult to share, and we want to apply them to data from a different site without necessarily having to transfer the data, because we might not have the permission to do that. So being able to transfer the parameters from one site to another in a federated manner is very important as well. The last thing I'd like to say is that all of this, apart from Johanna's stuff, which she's already commented on, is available in the toolbox that Sage is going to demonstrate. If you're inspired by some of this work, please get in contact. We are taking on both PhD students and postdocs. So if you want to come and work with us, please get in contact. With that, I'm going to turn it over now to Sage, who is going to continue the discussion with a practical demonstration.
Hi, I'm excited to show you guys some of the stuff I've been working on. So we've had an awesome, first of all, I'm Sage Rutherford. I'm a PhD student in Andre's lab in Nijmegen at Donders, but coming from Ann Arbor, Michigan. If you want to apply to the lab and want to talk about moving to the Netherlands, feel free to reach out. I'm happy to share any useful tips. So I've really been working on documentation and how to, there's a lot of diverse people in neuroscience research that come from,
you need to change this to the screen.
this one? Are we
No,
sharing it?
you'll have to start it again because you're still sharing the PowerPoint.
How do I change it?
Yeah, that's it. Sorry.
Loading.
Yeah, and now you can go in there, and then you can get this one.
Okay, perfect. Live screen. So I have been working on, let's skip that, on a lot of documentation there's a lot of diverse people in neuroscience, and let me get back to this. That, some people maybe come from a more like biological background and, maybe study neuroanatomy, where there's some people that are coming from like maybe a physics and mathematics background. And sometimes, the field moves really quickly and we don't always communicate that well with one another. And we can kind of, develop all these new fancy methods. And a lot of times they aren't able to really, people don't start using them because there's not clear documentation. So that's something that I call research debt, and I really care about working on it. And it's very important to have innovative methods and stuff, but it's also very important that people, lots of people in the field know how to use them. So our code is shared on GitHub on the PCNtoolkit, but we've also just recently added a new documentation page on Read the Docs. So you can find it just by clicking on this button. We also created a chat on Gitter, so if you have any questions about it, you can either open an issue on GitHub or you can chat with us on Gitter, and we'll try to answer some of the questions, whether it's just like, more high-level questions about how should I use this in my data, or very specific like implementation. I think we care about helping you use these methods because we really think some interesting science will come from them. So you can find the links to them on the GitHub. And yeah, we have just a few people there so far. So we'd love to see people from outside of our lab start to have conversations about normative modeling. I want to go over the documentation that is now on Read the Docs. So we have a guide for how to install, whether it's on just your own computer or maybe it's on kind of like a shared resource. It's very Python based. So I think using Anaconda is a great way to set up the installation, and having a virtual environment so that all the packages that are required for the PCNtoolkit don't get in the way of some of your other projects. And directions for all of these is on this page. We go into a little bit of background. I'm not going to talk about this today because Andre just talked about a lot of that. There's also an area where you can look at the functions and classes, and that kind of goes over what is the input to each and what are the parameters and the outputs that things return. There's a lot here, so I'm not going to go through everything, but I really encourage you to check it out. My favorite part of this page is the tutorials link. So as we mentioned, there's a lot of different algorithms that you can use to implement normative modeling. Gaussian process is one of them, Bayesian linear regression and hierarchical Bayesian regression. So we've created tutorials that really walk you through how to, from start to finish, run each one of these analyses. And there's links to the code on the Read the Docs, but then we've also included links for running it in Google Colab, which if you haven't heard of it, it's kind of like a cloud-based Jupyter notebook for running the code. So if you're interested in just kind of like running through it really quickly and don't necessarily want to actually run the cells, you could look at these on the Read the Docs and just kind of scroll through and see the steps that are involved. But if you actually want to run the code yourself, you can click the link to open in Google Colab, and I will walk you through one of the tutorials right now. I'm not going to run it because that would take a little bit of time and maybe be boring to you guys, but I'll just go through some of the steps so you can really start to see how everything's there, and you can kind of get a hands-on feel for what's happening in this. In this tutorial, we're using Bayesian linear regression, and we're using a couple of open data sets, the Human Connectome Project, the CAM dataset, and one other to predict cortical thickness. So we start by installing the PCNtoolkit. We then have to prepare our covariate data. There's kind of a set format that the algorithm expects your data to be in. So we go over that a little bit in the beginning. And again, all of the data is provided for you. You could, if you really want to try this out on your own data, you could substitute in your data here, but just make sure it's in the correct format. But if you're really just wanting to run it, everything is here. You don't need to bring your own data to it. So we format the covariate data. Here we're just using age, sex, site, and Euler number, which is a metric that represents data quality. We then have to prepare some of our brain data and merge together the brain and covariate data so that we make sure that we have all the same subjects and that there's no error in merging, because that would obviously mess the model up. We then go on, this is all just about kind of plotting the Euler number and excluding any bad data that has a very big Euler number. Combining them, again, just some formatting. And then at the very end is going over how to choose which ROIs you want to run it for. That's one of the great things about our method, is that it's not just like running it for, total gray matter volume or total brain volume. We really do like a very specific model for each part of the brain. So in this tutorial, I've selected just a few regions of interest. You could, we're using one of the free surfer ROI atlases, so you could change these to include other ROIs that you're interested in. You could add on or change them. And then it goes over how to include a basis expansion. Don't want to cover that too much because that might be kind of boring for this tutorial. And then we actually go over how to loop through and run the code. So, and at the end of it, we go over a little bit how to interpret what is coming out of the model. So there's a lot of numbers and a lot of things to interpret. So provided some links for learning a little bit more about them. Also in the read the docs, I think we're going to, it's empty right now, but we'll have a glossary that will kind of go over some of, how to unpack the output and interpret it. And in another one of our tutorials, we've included, it's not in the Bayesian linear regression tutorial, it's in the Gaussian process. At the end of the tutorial, we provide code for how to create these nice centile maps. So that was something that took me a while to figure out. So yeah, definitely reuse our code and don't reinvent the wheel yourself. So yeah, those are some of our resources. Hopefully you can get started running normative models. And also on this page, we've linked to some of our papers. I think we need to update this maybe with some of our 2021 preprints that we've linked into the chat. But this is kind of our homepage for where you can access all of our code and tutorials. And so you can learn to run these analyses yourself. And also there will be a page on updates. This documentation is a little bit of a work in progress, but this is kind of the homepage where you can learn how to run a normative model. So yeah, hopefully we can open it up a little bit to some questions. There's still like 15 minutes left. Maybe I will stop sharing and we can see the chat. Okay, oh, Richard linked in to read the docs. Yay.
Okay, are there any questions? If you have, put them in the chat or send them through. Thanks, Sage, for that presentation. And I'd just like to really emphasize again that this has really been, the product of a lot of work from, all members of the team, and there's really a lot of, it's a very active development arena at the moment. So as new data sets, new requirements arise, we respond to them.
If there aren't any questions, we could also maybe, why are you not using UK Biobank?
We are using UK Biobank, Fidel.
In one of the data sets that I created, it just creates this like huge, it just looks different than all the data sets. So I actually sampled and just randomly took like 10% for it.
The main reason why we can't just use UK Biobank is we're interested in a lot of psychiatric disorders, which have an onset earlier in life. So if we are sort of in the aging range, then UK Biobank is an excellent resource, and a lot of the work that Charlotte's been doing on validating against different sort of IDPs, image-derived phenotypes, has been done on UK Biobank. But we also are interested in what happens in youth. So UK Biobank is great for a lot of the stuff. But we still need other data sets as well.
Yeah, it's not quite a lifespan data set.
Yeah. Are there any other questions? If not, we could perhaps just discuss a little bit where we see this approach going in the next years. Walter. Great. Are you planning to publish some contributing guidelines? It would be great to be able to contribute somehow to the tool. Yes, that's something that we will definitely work on. The development of an API has been a discussion point in the group for quite a while. So yeah, that's something that we're aiming to do.
And we, on the PCNtoolkit GitHub page, not the read the docs page, but on the actual GitHub, there is a how to contribute guide that we kind of copied the template from fMRI Prep in another project, and yeah, always open to pull requests.
Yeah, you won't find it just yet. Also, we'll be aiming to get that coming soon.
It's on the development branch.
Any other questions? Great. If there are not, I think we can maybe just talk a little bit about some of the future directions. We've had lots of applications in the last year, some excellent work by Mariam and Thomas and Thomas Wolfers, and just really, and that was sort of the stuff, the work that I showed very briefly in terms of understanding individual variation and how it may or may not be the same as group differences, and then using this tool as a basis for subsequent classification. So you have these individual Z statistics. If you were to combine data from different modalities, so for example, if you're combining cortical thickness with, I don't know, brain volume and from some structures or something, they're completely different units. So if we have a common reference model that describes, for example, lifespan variability, and then we rescale all of the clinical data to the centiles defined from the same reference model, we have a common space with common units that I think Mariam's most recent paper shows that it provides a very interpretable and a natural basis for subsequent stratification. So that's one area that we're working on, is to really sort of to provide those kind of reference models that define a latent space for subsequent analyses in a very fine-grained, very comprehensive manner across, as high resolution as we can get to, so even down to the vertex level or the voxel level, and then working on how we can integrate those kinds of patterns of deviation, so we can say that someone's there's an individual fingerprint that might be different in different individuals. How does that reflect? How does that map on to, for example, genetics or subsequent other kinds of phenotypes? How does it give rise to symptoms? Can we use these kind of tools as an early detection system, so we can say that, particularly with longitudinal data, we have the ability to really get down to make very precise inferences as to when an individual might be deviating from an expected trajectory, and the idea would be that would be more sensitive than just looking at the data without recourse to the reference model that we would be using.
And something that when I started doing normative modeling that didn't initially make sense to me, you get the Z scores out as a text file, but they can be put back into brain space, visualized there, and then they can also be used as features in a predictive model. You might need to do some feature selection step like a clustering or a dimensionality reduction. But I think a really interesting, we've done the normative modeling, then what? How can they be used to predict behavior or look for, I'm trying to avoid the going back to group differences on the z-scores. It's a hard paradigm to get rid of. But in predictive modeling of behavior, I think that's a really interesting avenue that we'll see more of.
Yeah, so one question that I have, or like that I'm at the moment like a bit stuck with, is like you said, like this dimensionality reductions from these many z-scores that we get, and then like kind of correlate or relate them to like clinically like relevant information. Like how exactly would you do that? Like, do you have any like specific idea?
Got some good ideas about that. And Mariam, would you like to say something about that?
Yeah, I think so. The thing is, with when we are looking at the one-by-one mapping, right, from behavior to brain features. I mean, we shouldn't think about this mapping, in a traditional way. So right. So and when we are using this dimensional reduction of the score, we shouldn't just looking at the first order correlation, right? So you want to have the second, like you want to see how they are changing. Like you want to see the correlation between variance, like the alterations and the behaviors, right? So there isn't really a linear relation there. So this is how I interpret this behavioral and z-score mapping.
If I could add to that, the last question, yeah. So just very briefly, I think it provides a very rich space for subsequent stratification, and there are lots of different ways to do that.
Brain basis set.
Brain basis set is one, but also looking at latent variable models and finding overlapping representations within the same brain tissue. So there's a lot of work to be done in that space. I think we're out of time now. I would like to thank all the participants in this forum. I hope it was useful. I would just like to thank everybody really who helped.
It was fun.
It was a lot of fun. I'm sorry about having to swing the laptop around. We had some technical issues. And I would just invite everybody who's interested to get in contact with us. On Gitter, yeah.
Or GitHub.
Whatever you like. You can send a homing pigeon if you want, but we'd like to hear from you. We'd like to see more applications using this kind of software. I hope the next OHBM we can talk to you about the further developments of this kind of analysis approach.
Bye.
Bye-bye.
Bye-bye.
Thanks for joining in, everyone. So I guess we'll see you in 10 minutes, or some of you, in the next session, if you want to stick around. But thanks for the presentation.
Thanks a lot. Thanks very much for assisting us.
Cheers.
See you later.